Thank you everyone for joining us and welcome to the Cancer Research Institute's webinar series, Breakthroughs in Cancer Immunotherapy. Today's date is Tuesday, May 12th, 2015, and the title for today's webinar is Melanoma Immunotherapy, What's Next for Patients? Today's webinar is made possible through the generous support of Amgen. Amgen focuses on areas of high unmet medical need and leverages its biologics manufacturing expertise to strive for solutions that improve health outcomes and dramatically improve people's lives. A biotechnology pioneer since 1980, Amgen has grown to be one of the world's leading independent biotechnology companies, has reached millions of patients around the world, and is developing a pipeline of medicines with breakaway potential. Today's webinar, we will be live tweeting from the @Cancer Research Twitter handle using the hashtag CRIWebinar. We encourage you to join the conversation online. My name is Brian Brewer, and I'm Director of Marketing and Communications for the Cancer Research Institute. Cancer Research Institute is the world's only nonprofit organization dedicated exclusively to harnessing the immune system's power to conquer all types of cancer. We fund scientists around the world whose work has led to significant breakthroughs in treating cancer with immunotherapy. Over the next 45 minutes, you'll have an opportunity to hear firsthand from an immunotherapy expert how cancer treatment is undergoing a revolution thanks to these breakthroughs. After a 25-minute presentation from our speaker, we will open the discussion to questions submitted by you. You can pose your question at any time by typing in the Q&A box on your screen. Today's webinar is being recorded and will be made available via the Cancer Research Institute website and our YouTube channel. Now it's my pleasure to welcome today's speaker. Dr. Antoni Ribas is a professor of medicine, surgery, and molecular and medical pharmacology at the University of California, Los Angeles. As a physician scientist, Dr. Ribas conducts research focusing on checkpoint blockade, including CTLA4 and PD-1 blockade, adoptive T-cell therapy, and targeted therapies involving the BRAF and MEK proteins. He has published more than 200 manuscripts on laboratory and clinical research in melanoma. Dr. Ribas, welcome.
Thanks, Brian. It's my pleasure to be here, and I want to first thank the Cancer Research Institute for putting together this seminar series and inviting me to be part of it, and for all of what they do to support cancer research and implementation of immunotherapies for cancer. In 2013, at the end of 2013, the scientific journal that's on the top of the field, is called Science, looked at all of the field of science and said, what is the biggest advance that we're seeing? And they concluded that the biggest advance was cancer immunotherapy. And they concluded that for two areas, the advances in checkpoint blockade and the advances in adoptive cell transfer therapy. That's what I'm going to be focusing on in this talk. The whole premise is that there are cells of the immune system called the T cells or T lymphocytes that have the ability to specifically recognize and attack cancer. And they do that by having receptors that just recognize that particular protein. These cells need to be turned on to attack the cancer, but our body has developed a whole bunch of systems that are not prone to the immune system cell finding the cancer cell, because this is a system that has been generated to fight infections as opposed to fight cancer. But we've known for a long time that in some patients it may work. So for many years we were trying to turn on these cells, and we would do it by a series of ways, either doing some kind of a vaccine or giving cytokines like interferon or interleukin-2, or we've gone up to interleukin-21, trying to turn on the immune system cell, the blue cell, to attack the brown cell, the cancer cell. This worked, but just occasionally. It worked in a minority of patients. The biggest limitation is that we were not addressing the checks and balances that the immune system has. Those checks and balances can be positive by having molecules on the surface of immune cells that are modulating how that cell will interact with the tumor. And then negative checks, which are limiting the immune system to attack its target. It turns out that cancer hides behind these negative regulators called CTLA4 and PD-1, which is what we're going to be talking mostly about. And once we release those, then we can have the immune system attacking the cancer. There's another way that we could do this. We can generate an army of immune system cells and use them to give back to patients in what we call adoptive cell transfer therapies, or ACT. So on top are all of the approaches of active immunotherapy, vaccines, cytokines, immune modulating antibodies, including checkpoint inhibitors. And on the bottom is either cloning the T cells or making a large army from one cell, or genetically modifying the immune system cells to give them a receptor that's specific for the cancer, which can be a T cell receptor, or TCR, or a chimeric antigen receptor, or CAR, and that's by genetic engineering, and then adoptive cell transfer, or ACT. We'll start by inhibiting the negative regulators, CTLA-4 and PD-1, which is what's most broadly applicable in patients with advanced cancers. And it's been approved in patients with melanoma, and PD-1 is now approved in some patients with lung cancer. And the whole premise of the immune system is that it has a very specific way of recognizing the bad guys, the cancer cells. And I'm going to use the analogy of turning on a car, which is something that I took from slides that were generated by Jed Wolchok, a colleague of mine from Memorial Sloan Kettering. And Jed had said that the specific recognition of the cancer by having an antigen or a protein that's recognized differently from the normal cells to the cancer cells, it's like having the keys to your car, and you go to the parking lot and you'll only turn on your car. You will not turn on the car that's parked right by yours. And we want to turn on specifically the cell that's specific for the cancer. But once we do that, then we have to not only turn on the engine, but hit the accelerator. And that's called co-stimulation in the immunology terms. But once you hit the accelerator, very soon after you want to have a brake. And that's called checkpoint in immunology terminology. And the checkpoint blockades, like CTLA-4 blockade, is turning off the brake, so then the immune system can be turned on and not being actively turned off as it is usually when trying to fight the cancer. By doing this, we can have durable responses in a subset of patients. This is a manuscript that includes over 3,000 patients treated with one CTLA-4 blocking antibody called tremelimumab. You can see that this is the curve goes to 1 year, 12 months, 2 years, and then goes up to over 5 years. You can see that unfortunately in the first 2 years, the majority of patients that are not going to make it through this cancer are dying then. But the ones that make it to 2 years, they're very likely to live 5 years, telling us that with this approach of releasing the CTLA-4 brake, we get a subset of patients that have a durable tumor response that can last years. I treated my first patient with ipilimumab in 2001. She was a patient with lung and liver metastases, bulky. She got progressed on everything we had at that time. Now we're in 2015. It's 14 years later. I just see her once a year. She's gone on with her life. She just was treated at the beginning for the first year and then stopped and has not heard back from the melanoma. That's what we want to do in more patients. We'll get to get more patients to have these durable responses. So CTLA-4 blockade with ipilimumab and another one that's on clinical development called tremelimumab can do this in a subset of patients. And those, at the beginning, we called them miracle survivors, like these two patients of mine that were chronicled by Forbes. And in the front page said miracle survivors with different patients. There's a patient of mine who was a school teacher that I treated in 2003, and then another one who's a physician from New Zealand that I treated in 2004. And he would come from New Zealand, get the infusions, fly back. And now they're both going on with their lives, and the physician from New Zealand is inviting me to give a talk in November. That's going to be over 10 years after we first treated him. But the problem is that this only happens in a minority of patients. And I'm putting this complicated cartoon where on the left side we see where ipilimumab or CTLA-4 blockade acts, which is when the immune system is being turned on by the generals to the immune system called the dendritic cells. And the fighters, called the T cells, are turned on, which is the blue cell. And then that cell goes around the body and finds the bad guy, the cancer cell, that's in the tumor. The immune system regulates this very tightly. And on the activation step, where CTLA-4 blockade works, it has CTLA-4 is limiting the number of cells that the immune system allows to be activated. It's like seeing a James Bond movie where at the beginning James Bond is being told the bad guys are these guys and they have all these weapons, and here you have this little gadget that will help you fight the bad guys and find them. But you don't want that many James Bond around because there's a lot of collateral damage in all those movies, and there's a whole bunch of cars flipping over and a whole bunch of things. And that collateral damage in immunology would be called autoimmune disease, which is we have too much immune activation, then the immune system attacks normal organs. So that's why CTLA-4 is there, and we release it by PD-1, by ipilimumab or tremelimumab. So once the immune system gets to the tumor, then there's another way that the tumor protects itself. It's with through the expression of PD-L1 that turns off the fighter cells, the immune system cells by PD-1. We'll focus on that, and I'll focus on that because the treatments that are blocking PD-1 are advancing rapidly, not only in melanoma and in lung cancer, but I hear that they're being tested in 30 other histologies or different types of cancers. Here are CAT scans of a patient of mine that I treated in 2012. On the left are the CAT scans when he started, and the black part is the normal lung. On the top is this big mass that's on the right side of the that's obliterating all of the right side of the lung. You can see disappearing and becoming black after therapy. On the bottom, on the left, there's the liver, and there's a darker part, which is melanoma in the liver. Very bad disease. This patient was progressing on biochemotherapy, high-dose interleukin-2 and ipilimumab, so had no real options of treatment. And you can see afterwards that the light gray of the liver is what is mostly on the scan, and only a smaller part has the darker gray. That's in 2013. The patient, I saw him yesterday, so he's now in 2015, so it's 3 years later. I'm going to show you a picture of this patient because actually he sent me this picture. There's his email is on the bottom. Says Tom says, "Hi Tony, you're welcome to share this picture," and it depicts a patient that's really sick, that had lost weight, had fell on the floor, and he tells me, "I don't even remember falling and hitting my head." He was too weak to really get off his chair, and that was just a couple of weeks starting on pembrolizumab, on the PD-1 antibody, because he started very late on his course. But his immune system was ready to attack the cancer, and it worked for him. And you can see this picture of Tom with me on a promotional pamphlet that UCLA generated, saying that we're making progress on treating this disease, and we're making progress by harnessing the immune system. We gave to Tom an antibody that doesn't kill any cancer cells. It doesn't touch a cancer cell. If you put it on a test tube, the antibody, so the treatment, this pembrolizumab, with the cancer cells, it will not kill any. You need the immune system. So it was Tom's immune system that attacked the cancer and brought him from what he looked 3 years ago to a guy who now plays tennis every day and walks and does more exercise than I do. This is not just one-off. This is a series of patients. Here's a graph that we call a waterfall plot. The middle line is where the tumor sizes start. The bars that go down is a decrease in size by percentage, so from 100% to less than 100%, and going up are the patients whose tumor progresses. You can see that 2/3 of the patients are on the side of tumor shrinking. So we give an antibody that releases a break to the immune system, and 2/3 of the patients have some type of tumor shrinkage. And it doesn't matter if they have previously received ipilimumab or not. But this is not the only exciting part of this. The exciting part is that once we get the immune system to attack the cancer, it's likely to be a durable benefit. And this plot that we call a swimmer's plot, every patient is a row, and at the end you see the ones that have a little white arrow. The ones that have a white arrow are the patients who continue to respond. And at the time that we reported these scores, it was between 6 months and 18 months or a year and a half. And nearly 90% of the patients that had a response continued to respond throughout this time. Usually we have to talk about what are the drawbacks of this kind of therapy. And the drawbacks are not that many because the side effects are minimal. This is a busy graph that shows the toxicities, different types of toxicities. We can look at the bottom on the right that says total number of toxicities, and we focus on the column that says grade 3 to 4 toxicities. Those are it's 12%. So it's only 12% of the patients that are clinically relevant toxicities. The great majority of the other patients have no toxicities at all or very minimal. Occasionally, patients develop fatigue, skin rash, and diarrhea. Some patients develop thyroid problems. There have been patients that have had liver and kidney problems. They're usually less than 1%. It's something that we need to be aware of and we need to be watched, but this is not a toxic form of therapy. And it's not only this particular antibody pembrolizumab, the same data is generated by another antibody called nivolumab that it's been on clinical development. One of the side effects that I find interesting for patients with melanoma is vitiligo or depigmentation of the skin. And here I'm showing 4 pictures of patients that have lost the pigmentation. And actually, it's a side effect that we welcome because it tells us that the immune system is trying to attack pigmented cells in the body. And as it's doing it, it's attacking melanoma and also the skin pigmented cells. But I'm showing this one because I think it's funny. This is a patient who had a birthmark in the face. You see it on the left. And this birthmark had been there all his life, kind of didn't bother him anymore. But as we turned on the immune system to fight the cancer, we got rid of the birthmark. The circle's a little bit up, but I think it moved a little bit, but you can see on the right-hand side that the birthmark is gone. So we can get the immune system to very specifically attack pigmented cells, and in this case melanoma, and attack normal pigmented cells in the skin. But we can similarly redirect the immune system to attack lung cancer, bladder cancer, Hodgkin's disease, and others. And that has led to the early approval of 2 of these PD-1 antibodies, pembrolizumab with the commercial name of Keytruda and nivolumab with the commercial name of Opdivo. They're both approved for melanoma, and I should have updated these because since February of 2015, Opdivo is also approved for the treatment of metastatic squamous cell carcinoma of the lung. A huge advance in the treatment of this disease, and we look forward to a lot more and a broader approval of these agents and several others that are being developed that are blocking PD-1 or PD-L1. The data in scientific journals is being published in the highest journals. Here we have the New England Journal of Medicine, the highest impact medical journal, with studies with nivolumab, with this big separation of the curve between the yellow curve, which is nivolumab, and the old chemotherapy drug called the carbazine, being much better to be nivolumab. Then these other ones that have the blue and the yellow curve are pembrolizumab, and the gray curve is ipilimumab, which was the prior standard of care. Ipilimumab already improves overall survival for patients, but with the PD-1 antibodies, we improve it further. And I think we're going to see even more data like this in the near future. But the key thing is understanding why does this work, and why does it work in some patients? How does it work? Because with that, then we'll be able to make it more broadly applicable to more patients. Now, many of our groups have been looking into tumors of patients, and I want to thank the patients who go through biopsies as they're getting these therapies, because they're really allowing us to get a new level of understanding of how their immune system is attacking the tumor. And here you have my depiction of what we see on baseline biopsies. In some patients, we see the blue cells being the immune system cells that are mainly excluded from the tumor. Some of them are inside. But once this is there, it tells us that there's an immune system that's been trying to attack the tumor. And that immune system is made up by these T cells that have a specific receptors that recognize the tumor. And as they recognize the tumor, they make proteins called interferons. But also the same recognition process turns on the negative regulators like PD-1, the receptor that turns off the T cell. and interferon allows the tumor to hide itself and express PD-L1. So in a way, the immune system is trying to attack the tumor, and the tumor says, don't kill me. I'm going to protect myself by expressing PD-L1. So once we recognize these, then we can think, well, what we need to do is to release this negative regulator. We don't allow the tumor to hide from the immune system. We don't allow the immune system to get signals to turn itself off, so then the blue cell can then attack the brown cell. And once we do this, we release this negative checkpoint, and then the immune system cells proliferate, and I put them with these, they're bigger, they have this red nucleus, and then invade into the tumor, and that leads to the tumor shrinkage. So it's like having a pack of dogs that are ready to go and hunt, but they're all kept on leashes somewhere, and then we just release them and they can go and fight the cancer. And that's what we want to do, more patients. But it will only work by itself once we have the immune system, which then allows us to think which patients are benefiting to these therapies and which patients are not. And then it allows us to then think, what should we do about it in the patients that do not have benefit? And here is two kinds of patients. On the left, there's a patient that has CD8 cells, which are the immune system cells, has PD-1 and PD-L1, and that's a patient who goes on to respond. And on the right is a patient who goes on to progress. There's no brown staining, so there's no CD8 cells on the top right-hand side. There's no PD-1. There may be PD-L1, but if there's no immune system cells there, there's nothing to attack the tumor. So by recognizing this, then we think we can understand which patients are going to respond or not. And this is data that we generated with my colleague Paul Thume by analyzing a series of samples, studying what is required to get an immune response by blocking PD-1, and then how can we understand this and then predict the patients that we don't know. And we did that by asking samples from a colleague of us from Paris, Caroline Robert, who works in the Institut Gustave Roussy, and Caroline sent us samples, tumor biopsies from 15 patients. And Paul Thume, my colleague from UCLA, who's a faculty member in dermatology here, who have been studying all of these biopsies, looked at the biopsies and then quantitated the amount of immune system cells that are in the tumor. And we see from the top rows, there's the patient number 1, and we look at the first column, and it says number 58, that's a low level of immune system infiltration in the tumor. We look on the bottom, patient number 15 had 6,000 or 7,000 counts of immune system cells in the tumor. So the top ones, we said, well, those are patients that are unlikely to respond, so we call them, they're going to progress on PD-1 blockade. And the patients on the bottom, we say, those have a lot of immune system cells in the tumor. We think they're going to respond. And then we made this prediction and sent it back to Paris and said, could you unblind the series and tell us if the patients that we predicted progression, will they progress? And the patients we predicted response, will they respond? And here is the prediction, and on the right-hand column, it says clinical responses. You can see that the patient that we predicted progression, they progressed. There's one patient that we predicted response who progressed, and I think now with more refinement of the algorithm, we can even predict that one. And all of the other ones that we predicted response in the light blue responded. We were able to predict the outcome of 14 out of 15 patients just by looking at their tumor and seeing, are there immune system cells there ready to attack the tumor, and are they being blocked by PD-1? Which brings me to what do I think is going to be how we're going to treat in the future, which is in patients with any type of cancer, not only melanoma, we're going to be looking at their tumor. We're going to be quantitating how many immune system cells are there in the tumor. And if it's like the top part of this, which has a whole bunch of blue cells that are being kept in check, like those dogs in the car, we want to release them. And we unleash them by giving a checkpoint inhibitor. In this case, will be PD-1 or PD-L1 antibody. There may be others that will be added once we know more of what's keeping the immune system in check. But then there's going to be the cases where there's no immune system in the tumor. So now our job is going to be to bring immune system cells there. And one way to do that is to combine these PD-1 and PD-L1 blocking antibodies with other immune activators, like CTLA-4 blockade with ipilimumab, something that's moving very rapidly in the clinic, and is likely to become approved in the treatment of melanoma. But other immune system activators like cytokines and immune activating antibodies. We'll also be thinking about injecting things inside the tumors so we can change the microenvironment of the tumor. And there's clinical trials already ongoing on injecting one particular herpes virus called T-bag that can change the tumor microenvironment and bring in immune system cells. And then we can release them by releasing the immune checkpoints. Or we can do it with other more artificial approaches like we call TLR agonists and other ways of injecting tumors to turn their immune system. Most patients with cancer are not being treated with immunotherapies right now. They're being treated by chemotherapy or targeted therapies or radiation therapy. Well, there's a potential that those therapies could be used in combination with immunotherapies, and I think we're going to learn how to do that. But then there's going to be the cases where there's no immune system to be turned on. So we'll have to vaccinate the patients to turn on the immune system, or we'll have to genetically engineer the immune system to give them the receptors to recognize the cancer. You may have heard the excitement advances with chimeric antigen receptors to CD19 for patients with leukemias and lymphomas, which is advancing rapidly in the clinic with several companies developing those approaches, but also engineering the native T cell receptors or TCRs for adoptive cell transfer therapy. So with this, we're seeing then combinations that are already making it to the clinic, like the combination of a PD-1 antibody nivolumab with a CD4 blocking antibody called ipilimumab. You can see curves like this one also published again in the New England Journal of Medicine, where you can see this huge separation between the patients who had the treatment that we're all excited about, the PD-1 antibody alone is on the bottom now. We can see the combination is on the top. So very rapid progress. We're making these curves shift very rapidly. And what we are excited about, that's an improvement in the field, it's never good enough. We have to do better because we have to get this benefit to more patients. I think this is rapidly advancing in clinical trials. So I wanted to convey to you is that we're making a lot of progress in the treatment of melanoma and other cancers. Here's a conclusion about the advances in melanoma with BRAF and MEK inhibitors that's improving the lives of patients. But then we're getting into adding the immune system and taking the brakes on the immune system by releasing PD-1 blockade, which allows us to get a lot more patients that benefit long-term. And the patients who don't benefit long-term to this therapy, we're understanding what's missing. We're designing approaches that are combining and allowing to get the improvement further, like the addition of ipilimumab, targeted therapies on the patients who don't have an immune system that's going to be activated. We're going to do adopt. So with this, I'll be happy to take any questions from the audience or questions that may have been sent in advance. I'll turn it back to Brian.
Great, thank you so much, Dr. Thanks, Dr. Ribas. Thank you for that. We have gotten a number of questions from the audience, and I'm just going to get right into them because we have so many of them. The first one is, you mentioned toward the end of your presentation how you can use oncolytic viral therapy to recruit T cells to the site of the tumor. Can you talk a little bit more about this? Because recently there's been a great deal of attention through a 60 Minutes piece on using the polio virus as a cancer therapy, I believe in brain cancer. What sort of oncolytic viral therapies are available in melanoma today?
So the field of oncolytic virus therapy is based on viruses that can infect cancer cells. And sometimes they've been engineered to preferentially infect cancer cells and not normal cells, like T-bag, like vaccinia viruses, and like polioviruses. And they're engineered so they replicate preferentially in cancer cells. And then they kill cancer directly. But as they do that, they have viral antigens that are very powerful. And then they recruit an immune response. And then that immune response can allow to get the immune system activated for cancer cells that are not injected and are farther away. Otherwise, it would just be a local therapy, but it can be turned on as a systemic therapy by enlisting the immune system. But we're learning that the immune system has these checks and balances, the same ones that we talked about before, And by releasing those checks and balances, either CTLA-4 or PD-1, the hope is that we'll get these viral therapies to be even more efficacious and have activity well beyond where they're being injected.
So actually, this segues nicely into my next question, which is about combination immunotherapies, which you did touch on. So you can see a potential future of combining oncolytic viral therapy with checkpoint blockade. Are there any other types of combinations that seem very promising today?
I think the field is exploded with a whole bunch of combinations, and most of them are very well rationally designed, and they're addressing what are the limiting steps for single agent immunotherapy to attack the cancer. And they fall in these categories of bringing immune system cells into the tumors with a combination with ipilimumab or CD4 blockade or tremelimumab, combination with other immune activating antibodies to OX40, to CD40, to 4-1BB or CD137, cytokines like interleukin or interferons, combinations with the oncolytic viruses, and combinations with standard of care therapies like targeted therapies, chemotherapy, or radiation therapy. I think the next 2 or 3 years, we'll see a lot of these combinations, and we'll see that they work in some subsets of patients in different cancers. And I'm excited about the benefits that that's going to bring in a short period of time.
Great, thank you. So you said benefit in a subset of patients. What sort of work is going on today to help identify which patients are more likely to respond to an immunotherapy or not?
And I think this approach of diagnosing what's the interaction between the tumor and the immune system, and how the immune system is dealing with the tumor, and more importantly, how the immune system is not being able to attack the tumor and what's limiting, I think that will allow us to then personalize the immunotherapy and tell this patient needs this type of immunotherapy approach, and this patient needs this other type. And sometimes it's going to be single agent therapies because there's only one thing limiting, and sometimes it's going to be a combination of 2 or 3 immunotherapies or immunotherapy as standard of care therapies to get that immune system to attack that cancer.
I thought it was very fascinating. It made a lot of sense how you showed how the presence of CD8 T cells was a predictor of response to checkpoint blockade therapy. I thought that was very good to see. So let's say I'm a patient and I've been diagnosed with melanoma. At what stage in my disease will I become eligible to receive some form of immunotherapy, or are there other options that I should explore first?
So like many other treatment approaches for any disease, they're first tested in the more advanced disease, and these are being tested in what we call stage 4, or the cancer that has spread throughout the body and has made seedings in different organs. We're starting studies where we're trying to prevent cancer from going from a stage 3 or a cancer that can be taken up by surgery but has a high risk of coming back as a stage 4. We're trying to prevent that it becoming stage 4. But those are studies that take a long time because we need a large number of patients treated for a period of time and then followed for years, because not everybody that's being treated in that setting will develop a relapse of their cancer. So those are studies that will take longer, and they're only available. These agents are only available in clinical trials at that time. People may think, well, I should just get it outside of a clinical trial, but it's not always straightforward that something that's working in one setting will do the same in another setting. And it's because, in this case, we need to have the immune system interacting with the cancer. And if the cancer is not being active, then the immune system may not be interacting well. So maybe we don't need to release its brake. Maybe we need to do something else. I'm trying to play a little bit of devil's advocate because there's a reason why we have to go one step at a time and move forward by generating the right scientific data and then being able to advise our patients. But the good news is that even in the worst case scenario, when, in my case, for what I treat, melanoma has spread throughout the body, even in the bad areas, the bone, the liver, the brain, we're now getting a significant subset of patients to do well long term. And by long term, I mean years, not just a handful of months.
Let's say a patient is receiving an immunotherapy, such as a checkpoint blockade, and they may be responding well, and then the response stops and the tumor begins to progress again. What kind of options are available to patients in that situation?
I think from the scientific standpoint is understanding why that patient progressed in particular case, but it takes a long time and that's unlikely to be beneficial to that patient. But I want to again thank the patients who go through biopsies to allow us to study why is that happening. Because I help them, but it may help others for sure. What we're trying to think is what stopped working. If a patient responded for a period of time and then the tumor regrew, what happened there? There has to be something that changed. It cannot be just bad luck. It's something that we should be able to understand. And once we understand it, then we can give the right treatment. Unfortunately, for the first patients, it may be too late, but for afterwards, I think we're going to be making the right decisions on how to combine therapies in that case.
Great. So to switch gears here a little bit, let's talk about vaccines. And specifically, we have a question from the audience about someone who has had his or her melanoma surgically removed. There's currently no evidence of disease. Can a person like that consider exploring a melanoma vaccine to prevent recurrence? And if so, are there any studies like that ongoing, and what's been the results of those?
There's been a long-standing interest in vaccines, and there's been vaccine trials in melanoma for probably around 100 years. A lot of good ideas, a lot of things that were based on solid science that have been brought to the patients. Unfortunately, the field of vaccines has yet to live to its promise. There's been more examples of vaccines that didn't work at all compared to a sugar pill, or in some cases that have been even suboptimal, where the patients on the sugar pill group did better than the vaccine group. As it stands, there's no approved vaccine that has a track record to improve the outcomes of patients. That doesn't mean we should stop studying these, but it should be taken with a little bit of hesitance when I hope this patient is not being told at some place that you should pay for a vaccine that works, because there's nothing that works. If something works, actually ends up being approved by the FDA and then it's available to everybody. There's clinical trials of vaccines ongoing. That important would be for the patient to talk with their oncologist and try to see, is that vaccine any different from everything else that has been tested before? And if it is, then maybe there's a benefit to participate in a clinical trial. But again, the track record has not been good despite multiple attempts.
How different are melanomas from patient to patient? We hear that melanomas are often caused by environmental conditions, which can result in mutations leading to melanoma. Is each patient's melanoma different?
Absolutely. I've been treating melanoma for 15 years, and I think every one of them is different. And they're different not only in the way they behave, but when we look at their genes, each one is different. Some of them are driven by the same gene, like the BRAF mutation, which is in around 50% of melanomas. But that's just one mutation out of the hundreds of thousands that tumor will have. And chances are all of the other ones will be different from one tumor to the other. So it's hard to generalize on any aspect of cancer, on any particular melanoma. What's becoming exciting is that the more mutations that melanoma has, which we used to think that it was a bad thing, now we're starting to think that maybe it's a good thing, because immune system is more able to detect a melanoma that has multiple mutations and has a whole bunch of genomic alterations, as opposed to the melanoma that has less. where the immune system doesn't have that many things to focus on to differentiate from normal tissues.
We've seen that in some other cancer types as well, so it is a very interesting area of exploration. I'm wondering how close. So we mentioned looking at tumor tissue and studying the presence of T cells in there as a marker for or predictor of a potential response. But of course, tumor biopsies are rather invasive by definition, and it would be much easier and less invasive if we could develop a blood test to determine whether or not a patient would respond to immunotherapy. So how close are we to developing such a blood test?
For immunotherapy, I think we're a little bit far away because immunotherapy, it does, so the immune system cells circulate through the blood, but all of the action is in the tumor. The blood has to be a surrogate of what's happening in the tumor. Is the immune system recognizing and attacking the tumor? And I think we need to know a lot more about that to be able to make surrogates in blood. We've studied a lot in blood, and it's allowed us to learn a lot of biology on how the immune system behaves. But I think we're a little bit far from having a blood test that will diagnose how the immune system interacts with the tumor and will it be likely or not to respond to one type of therapy.
Okay, I'm just going through the questions from the audience here, and we have another one about another type of vaccine, a DNA vaccine. And the question is, where do DNA vaccines stand in the context of combination treatments with immunotherapy?
So DNA vaccines have been tested for several years, probably around 10 to 15 years, and the idea is that you inject something into the tumor and that changes the tumor in some way, gives it another gene, and then becomes either altered in a way that the tumor dies or enlists the immune system. There's DNA vaccines that may have a use now that in combinations because they may allow to alter the tumor and make it more immunogenic or more visible to the immune system by providing a gene that attracts immune system cells. I think there's going to be quite a bit of more research in that. Conceptually, it's not that different from what we were talking about before the oncolytic virus therapy, where we're injecting, the virus brings DNA that becomes immunogenic and enlists an immune response. So I think there's two different ways of trying to achieve the goal.
Okay, and just to wrap up here, in general terms, I'm hearing a lot of optimism, a lot of good news, not only for melanoma patients, but also for patients with almost any type of cancer, thanks to immunotherapy. Can you just say a few words about where we are today in relation to where we were even just 5 years ago? And where do you see the future? What will the next 10 to 15 years look like for patients?
I would say not even 2 or 3 years ago, there's been a revolution in the treatment of melanoma with the application of these immunotherapies that are leading a subset of patients to have durable responses. But this is just the start. We are now understanding the rules of the game. What do we need to do to the immune system to attack the tumor and do it in a sufficient number of patients that we can understand exactly how it works. That's allowing then to think of what is not working in other patients, or how can we apply these from melanoma to other cancers. And we're also seeing a very rapid process where these immunotherapies are being developed in different cancers. I think in the future, maybe 3 to 5 years from now, immunotherapy may be the frontline therapy for the great majority of patients with cancer. And it's going to be guided by understanding, is the immune system there trying to attack the tumor and what's limiting it and how can we release that? Or can we enlist the immune system to attack the tumor if it's not trying to do that? And how can we take out the different breaks that are not allowing it?
Well, thank you very much, Dr. Ribas, for this. This was very enlightening, and it's good to hear so much positive results from the research being conducted around the world, including research funded by the Cancer Research Institute on this very promising area of cancer treatment. I'd like to thank our friends at Amgen once again for their generous support of today's webinar. And also to let you know that June is Cancer Immunotherapy Month, and we've developed a number of very exciting, informative programs throughout the month designed to increase awareness of immunotherapy as well as to raise funds for the work of the Cancer Research Institute. So first, I'd like to point out our next set of webinars, which we're doing as part of Cancer Immunotherapy Month and ramping up toward the month. Our next one will be on May 20th at 3 p.m. Eastern Daylight Time with Drs. James Gully and Ravi Madan of the National Cancer Institute, where they'll be talking about emerging concepts in prostate cancer immunotherapy. These webinars are free, of course, and will be available after these dates. So if you happen to miss them, please come to the Cancer Research Institute website to view them. And you can do that by going to cancerresearch.org/webinars. Also, during the month of June, we are going to white out cancer on June 12th. And the way this works is you wear something white on June 12th. You take a picture of yourself, or you have a picture of you taken with a group, and you share it via social media using the hashtag #WhiteOutCancer. And we're really looking forward to this as a way to increase awareness for the need for more research funding around immunotherapy. Also, for those of you who are interested in learning more about immunotherapy, and in particular would like to either contact patients who've been treated with immunotherapy, or if you'd like to learn about clinical trials of immunotherapy, we encourage you to go to our website at theanswertocancer.org. This is a patient and caregiver-focused portal that the Cancer Research Institute has designed to provide friendly, accessible information about all the cutting-edge breakthroughs like the ones you heard from Dr. Ribas today. And with that, I'd like to conclude today's webinar. I'd like to thank everyone for attending. And of course, Dr. Ribas, thank you very much for your time. And we hope to see you all at our next